covered DNA structure, replication, repair, and recombination in the last few lectures. Clearly, all the developments around DNA are a pretty big deal. Proteins at the start of any consideration of metabolism get a similarly large amount of attention. The structure of proteins, their functions, their importance, and how they're controlled. Again, a pretty big deal. In this first lecture focused on RNA, we're going to begin connecting the immensely important topic of DNA with the immensely important topic of proteins. Now, RNA is much more than simply a copy of the DNA blueprint. RNA not only carries the message to make a protein, but it also provides the decoding rings necessary for converting nucleotide sequence to amino acid sequences in proteins. And RNA is an even more important component of the ribosomes that actually make the proteins. In this lecture, we're going to focus on the synthesis of RNAs, specifically how RNAs are made from DNAs. First, we'll discuss similarities with DNA replication and also how it differs. Then we'll examine how RNA polymerases copy DNA to make RNAs. In this lecture, you will also learn how human cells shuffle their genetic deck of cards to make about 100,000 different proteins, using fewer than 30,000 coding sequences. You'll also see diseases arising from poorly shuffled decks. And last, we'll talk about how our knowledge of transcription that occurs after death helps forensic scientists establish the time of death accurately. So before we get started, let's make sure we're on the same page with respect to nomenclature. We use the term transcription to indicate the reading of DNA sequence to make RNA. Transcribing literally means writing across, in this case going from DNA over to RNA. Second, regions of DNA that get transcribed into RNA are called genes. Now, some people use genes in a narrow sense to indicate only the regions of DNA that code for protein. Because we're turning our attention to RNA, we will use a much broader definition. Genes are DNA sequences that get copied into RNAs. Copying of RNA is catalyzed by an enzyme called RNA polymerase. Unlike DNA polymerases, cells don't have so many different kinds of RNA polymerases. Prokaryotes only have one, while humans have three. The three eukaryotic RNA polymerases are given Roman numerals 1, 2, and 3 to distinguish them. Transcription, like DNA replication, involves building a string of nucleotides across from a DNA template using the base pairing rules. Of course, to build an RNA, cells use RNA nucleotides rather than DNA nucleotides. Remember that RNA nucleotides are very similar to DNA nucleotides with two important differences. One is that the sugar in RNA nucleotides is ribose, not deoxyribose. The other is that instead of the bases A, T, G, and C, RNA uses A, U, G, and C. U pairs with A just like T. Because you're familiar with DNA replication, let's think about how transcription to RNA differs from copying DNA. First, when a sequence of DNA is transcribed to RNA, only one of the two DNA strands is copied to make the RNA. The DNA strand that is copied is called the template strand. The DNA strand that is not copied is called the non-template or coding strand. The coding strand has the same sequence as the RNA that is made, except that it has T's where the RNA, of course, has U's. Part of what makes our RNA-oriented definition of genes much broader is that cells make several different kinds of RNA. Not all types of RNA code for proteins. I think of the different types of RNAs as siblings in a family construction business. We'll consider three of them here. Tina, or tRNA, Mike, or mRNA, and Roberto, or rRNA. The RNA family business makes custom-designed proteins in a factory known as Ribosome Industries. The way it makes its products is a trade secret, so instructions are written in a code, the individual pieces of which are called codons. Mike's job is pretty simple. He's a messenger who carries the coded information from the corporate headquarters to the factory, where the proteins are built in an assembly line. Roberto is chief of operations at the factory and provides support to all the operations there. Without Roberto, the factory does not run. The most interesting role is played by Tina. Not only does she provide the information necessary for decoding the coded instructions that Mike is carrying, but she also ferries the raw materials to the factory so Roberto can direct their assembly into proteins. Okay, now that may be a bit silly, but it's reasonably accurate descriptions of the functions of the three main types of RNA. In our analogy, the corporate headquarters is the DNA. Mike in the RNA world is known as a messenger RNA, where the job is to carry the coded information to the ribosome. This information is carried in the form of groups of 3 nucleotides called codons. Each codon in the genetic code corresponds to a specific amino acid in a protein. Roberto is a ribosomal RNA, or rRNA. Cells have several rRNAs, and they, along with numerous proteins, self-assemble to make the ribosomes, which are protein synthesis factories in the cell. In a ribosome, rRNAs perform several functions, as we'll see in the next lecture. But one is to provide a scaffolding onto which the proteins attach to assemble into a ribosome. Since RNAs are single-stranded, they can fold into 3D structures that DNA can't. For example, a structure of the rRNA called 23S can fold on itself to make a sort of mold that ribosomal proteins bind to in order to form the ribosome. Roberto is truly central to the factory's function. We'll see in the next lecture that he even works on one of the assembly lines. Tina in the RNA world is a transfer RNA, or tRNA. This role is to provide translation of the genetic code's instructions in nucleotide sequences and convert them into amino acid sequences for making a protein. There are 20 different kinds of tRNA, each specialized to carry one of the 20 amino acids needed in the ribosome during protein synthesis. tRNAs read the codons in messenger RNA, and they also transport the amino acid corresponding to that codon to the ribosome factory. One last thing about the RNA family. There are also quite a few cousins in the family business, both small and large, that play roles in regulating the factory output of proteins. Now, you'll hear more about them in a later lecture. But whichever family member we're talking about, they're all made in the same way, by copying a region of DNA. So let's get down to making these RNAs. The synthesis process is essentially the same for all types of RNA. It is also the same as addition of nucleotides for making DNA. As with DNA replication, RNA synthesis only advances in the 5' to 3' direction. One important difference between DNA polymerases and RNA polymerases is that RNA polymerases do not require a primer to start making RNA. Once RNA polymerases are in the right place to start copying the DNA, they just begin making RNA by copying the DNA template. Unlike DNA replication, where every nucleotide of the parental DNA gets copied, transcription only copies selected regions of DNA. This, of course, brings us to an obvious question. How do RNA polymerases know where to start copying on the DNA? Consider the challenge. In a human cell, there are over 6 billion base pairs of DNA, and the average size of a gene is between 10,000 and 15,000 base pairs. An RNA polymerase must find the right place to start transcribing a gene, catalyze synthesis of the RNA, and then stop copying at the end of the gene. Finding the right start site for a gene in human DNA is equivalent to finding a 5-foot stretch on the 600-mile drive from Louisville, Kentucky to Washington, D.C. It would help to have a sign to mark the spot, wouldn't it? It turns out that cells do have signs for RNA polymerase telling it where to start and stop copying. The start sign is a sequence in the DNA called a promoter, and it is located adjacent to a gene. Likewise, there are sequences called terminators in the DNA that act as stop signals. The promoter is said to control the gene it is associated with. This is because RNA polymerase physically binds to the promoter sequence to begin transcription. If the RNA polymerase does not bind at the promoter, the gene will not be transcribed. We'll discuss the process of transcription first in prokaryotes and later in eukaryotes. Much of what we know about transcription in prokaryotes comes from studies of E. coli. Prokaryotes have a single RNA polymerase that transcribes all their genes. Remember that E. coli had only one DNA replication origin. By contrast, it has thousands of promoters, and their sequences are not identical. To understand a promoter, we must orient ourselves at the transcriptional start site, the place in the DNA across from which the first RNA nucleotide is placed to start synthesis. We call this location in the DNA the +1 site. This is equivalent to the starting point of a race. Orienting in the DNA this way, we see promoter sequences before the start site. This is like runners in a foot race who line up behind the starting line. The first of these promoter sequences is found at -10. It is a sequence of 6 bases, TAT, AAT, and it's found in the majority of promoters. There is some variation of this sequence from gene to gene, but TAT, AAT is called the consensus sequence. If we look further upstream from +1, centered at position -35, another sequence of 6 bases is quite similar for most E. coli genes. The consensus sequence here is TTG-ACA. The sequences centered at -10 and -35 are recognized and bound by RNA polymerase before transcription begins. RNA polymerase of E. coli contains several polypeptide chains. One subunit that's temporarily attached is called sigma. Sigma can attach to the -10 and -35 sequences in the promoter. The sigma subunit acts like an usher that guides RNA polymerase to its seat on the promoter. After transcription begins, the sigma subunit separates from the polymerase, which continues RNA synthesis. Meanwhile, the sigma subunit finds another RNA polymerase to escort to a promoter. Let's discuss what it means to have a consensus sequence at -10 and -35. I mentioned that they vary a bit. What does that mean? Well, if I align the -10 regions of 100 genes, I can determine the percentage of the time each base occurs from -15 to -10. For example, the -15 base is T 77% of the time, and each of the other bases only a few percent of the time. -14 is base A 76% of the time, and each of the others only a few percent each. The consensus sequence then is the most common base found at each position. But why aren't promoter sequences all the same? Shouldn't changes in these sequences affect how the RNA polymerase and sigma factor bind to them? They would, and that's exactly the point. The efficiency and tightness of the binding will vary as the sequence varies. If we have 2 sequences, one perfect for polymerase binding and one that's off by 1 base, the sigma would escort the polymerase to the perfect one more easily than to the less perfect one. The consensus sequence is the best sequence for binding. The more closely a promoter matches the consensus, the better the polymerase binds and the more transcription occurs from that promoter. The polymerase likes perfect seats better. Conversely, the more a promoter varies from the consensus, the less transcription is seen from that promoter. Is that a good thing? Well, the more transcription from a promoter, the more RNA copies of the gene are made, and the more RNA, the more the cell can make of the protein encoded by it. And why, you might wonder, would a cell want to make different amounts of various proteins? The answer is that protein synthesis is an energetically expensive proposition. Proteins should be made only when they're needed, and only in the amounts they are needed. And cells need different amounts of different proteins. So for example, a cell of E. coli only needs 2 copies of DNA polymerase III to support bidirectional replication from an ORAC. On the other hand, an E. coli cell may have upwards of 70,000 ribosomes, so it will need thousands of copies of each protein making up the ribosome. So it's natural and important that cells make more RNAs for ribosomal proteins than they do for DNA polymerase III. And at one level, it is the job of promoter sequences to determine how much RNA is made in cells. What happens after the polymerase has bound to the promoter? Well, once bound to the promoter, the RNA polymerase unwinds the DNA strands to create what is called a transcription bubble. In contrast to the bubble we saw in DNA replication, the transcription bubble does not have replication forks at each end. Unwinding the strands to form the bubbles is made easier by the fact that most promoters have a sequence rich in AT base pairs. Unlike DNA polymerase, RNA polymerase requires no primer to start synthesis. At first, the RNA polymerase simply strings together 8 or 9 nucleotides complementary to the template, while still sitting at the promoter. Then it begins to move along the template, unwinding the DNA ahead of it. No helicase enzyme is needed since RNA polymerase can handle the unwinding too. As it moves, RNA polymerase maintains a transcription bubble of 12 to 15 base pairs and synthesizes RNA complementary to the template strand at the rate of about 30 to 50 nucleotides per second. And behind the RNA polymerase, the DNA template is being rewound, displacing the newly made RNA from its template strand. Just as in replication, the opening up of the DNA strands for transcription creates overwinding ahead of the bubble. And just as in replication, DNA gyrase comes to the rescue to release the tension. As already mentioned, an RNA chain complementary to the DNA template is built by the RNA polymerase by joining of the 5' phosphate of an incoming ribonucleotide to the 3' hydroxyl of the last nucleotide of the growing RNA chain. How does the polymerase know where to stop? That's where the terminator sequence built into DNA gives the signal to RNA polymerases to stop transcription and dissociate from the template. In bacteria, if the RNA being made encodes a protein, ribosomes will bind to the starter end at the 5' end long before the entire RNA is made and released from the DNA. The ribosomes will begin translating the messenger RNA to make the protein it codes for, even as the RNA polymerase is still transcribing the rest of the RNA. Doing both at the same time is possible because in bacteria there is no nucleus, so transcription and translation occur in a single compartment. We've seen that once the RNA polymerase is positioned correctly, it strings RNA nucleotides together, moving down the template till it has synthesized the sequence called the terminator. The terminator sequence helps signal the polymerase to stop transcribing and release the newly made RNA from the DNA template. The process of making RNA is essentially the same in eukaryotes as in prokaryotes. The differences are external to the synthesis process. Eukaryotic DNA exists as chromatin, where the DNA is tightly associated with histones and other proteins, increasing the complication of the process. Such packaging of DNA must therefore be loosened to allow the RNA polymerase access to the template in the region to be transcribed. Now we'll talk more about these considerations in Lecture 32 on gene expression. A second difference in eukaryotic transcription is the presence of 3 RNA polymerases, not one as in bacterial cells. These different polymerases transcribe different subsets of RNAs. For example, RNA polymerase I transcribes the ribosomal RNA genes, while RNA polymerase III copies tRNA genes. The RNA polymerase we will focus on most is RNA polymerase II, which transcribes protein coding genes to make messenger RNAs. All 3 eukaryotic RNA polymerases need additional proteins to help them initiate transcription. In prokaryotes, RNA polymerase had the sigma factor helping it to find and bind to promoters. Additional proteins are needed by eukaryotic RNA polymerases, and they are referred to as general transcription factors. Finally, eukaryotic cells have a nucleus, and that is where transcription happens. RNAs produced there are processed further before they are sent into the cytoplasm. Now, protein synthesis or translation happens in ribosomes as in prokaryotes, but they are located in the cytoplasm. Eukaryotic promoters have some similarities to prokaryotic promoters, including a so-called Tata box sequence, about 25 base pairs upstream of the start of transcription. Eukaryotic RNA polymerases need general transcription factors to help the RNA polymerases find the promoter and initiate RNA synthesis. These transcription factors are named TF2A, TF2B, and so on, where TF stands for transcription factor, 2 stands for RNA polymerase 2, and the other letters distinguish individual transcription factors. Transcription in eukaryotes requires the general transcription factors and the RNA polymerase to form a basal transcription complex. The first step in the formation of this complex is the binding of the Tata box sequence by a transcription factor called the Tata binding protein, or TBP. When the binding protein binds the Tata box sequence, DNA bends at this point and takes on a structure suitable for the binding of additional transcription factors and RNA polymerase. The final step in the assembly of the basal transcription complex is the binding of a general transcription factor called TF2H. TF2H is a multifunctional protein that works as a helicase as well as a kinase. Now, the kinase activity of TF2H adds phosphates onto the tail of the RNA polymerase. This phosphorylation of the tail appears to be the signal that activates the RNA polymerase, allowing it to move forward and begin transcription. So in eukaryotes, the RNA polymerase is recruited to the promoter by a bunch of transcription factors that bind there first, and it's sent on its way down the template when it gets a nudge from TF2H, which phosphorylates the polymerase's tail. RNA synthesis proceeds as in prokaryotes. Though bacteria could begin using RNAs for protein synthesis even before messenger RNA synthesis was complete, several differences in eukaryotes preclude that possibility. The first reason eukaryotic messenger RNAs can't be translated as they're being made is that eukaryotic messenger RNAs are made in the nucleus, but ribosomes for translating them to protein are located in the cytoplasm. As a result, messenger RNAs must be moved to the cytoplasm. But before they get moved, messenger RNAs get trimmed and decorated in other ways. The first trimming process we will consider is called splicing. It only occurs in eukaryotes. In prokaryotic genes, the information for making proteins is contained in one uninterrupted block directly from the DNA. You might think of prokaryotic genes as TV programs with no commercial breaks. By contrast, eukaryotic genes have introns, or non-coding regions that interrupt the coding information, which is contained in segments that we call exons. A eukaryotic gene, then, is like a show that is broken up by commercials that have nothing to do with the program itself. We interrupt this exon to bring you an intron about something totally unrelated. If you wanted to watch the show without commercial interruptions, you would need to cut out the introns and connect the bits of the exons or the program together. And that's what plants, animals, and other eukaryotic cells do to their RNAs. Non-coding introns are removed before the messenger RNA is sent out of the nucleus to be used for protein synthesis. The whole process of removing the introns and joining the exons is what is called splicing. What is even more interesting about eukaryotic messenger RNAs is that some of the exons may also be removed from a transcript to make the spliced RNA. For example, in some cells, the primary transcript could contain exons 1, 2, 3, 4, and 5, but another cell might splice differently and make messenger RNAs containing 1, 2, 4, 5, or 1, 2, 3, 5. The individual exons and their overall order are still the same, but by leaving some of the exons on the cutting room floor, so to speak, it becomes possible to create messenger RNAs that will be translated to make slightly different proteins. This phenomenon is called alternative splicing, and because of it, we can make over 100,000 different proteins from fewer than 30,000 genes. Splicing is a double-edged sword. Though it enables cells to increase the kinds of proteins it makes from a given gene, Mutations affecting splicing can have profound effects on human health. It is estimated that as much as 22% of gene mutations affect splicing and cause diseases ranging from Duchenne's muscular dystrophy to Hutchinson-Gilford syndrome that leads to premature aging. Splicing isn't the only alteration eukaryotic cells make to their messenger RNAs. They also decorate them at both ends with special structures. The 5' end gets what's called a cap, which contains a modified guanine nucleotide. The cap protects the 5' end of the messenger RNA from degradation by nucleases since it alters the normal 5' end they would attack. The cap will also be important in protein synthesis, as we will see in the next lecture. Transcription of eukaryotic messenger RNAs does not end at fixed sites. Instead, a special "this is the end" structure is added before termination occurs. This is done by cutting the messenger RNA to create a new specific 3' end. Then an enzyme adds a tail of about 200 adenine-containing nucleotides to the new 3' end. This tail of A's is called a poly-A tail. There's evidence that this poly-A tail plays a role in efficient translation of the messenger RNA as well as in the stability of the messenger RNA. Together, the 5' cap and the 3' poly-A tail on the messenger RNA signal that processing of a messenger RNA is complete. At this point, the transcripts are described as mature, and the messenger RNAs can be moved to the cytoplasm where they direct the synthesis of proteins. RNAs are full of surprises too. It turns out, rather unexpectedly, that transcription continues after death. What? Yeah. How do dead cells do anything, let alone make new RNAs? Well, it turns out that you could be dead, no chance of revival, but your cells may not be dead, at least for several days. In 2016, researchers at the University of Washington discovered that hundreds of genes were transcribed after death in both rodents and fish. And this wasn't just a case of something that was already happening, continuing until it petered out. For example, organs intended for transplants can be removed up to 30 minutes after death. Skin grafts can survive if taken half a day after death. But that's not all we're talking about here. Rather, there were specific genes that began being copied into RNA after death. In some cases, transcription was still going strong 4 days after the death of the animal. Moreover, post-mortem transcription is not confined to rats and fish. Scientists have discovered similar activity in tissues from human cadavers. Strangely, some of the genes that were transcribed after death were developmental genes that normally are active only in embryos and are silenced after birth. Among the genes that began to be transcribed were some that promote cancer. This last observation may explain something that has been a mystery for years: why people who receive organ transplants from a recently dead donor have an increased risk of developing cancer. While much remains to be understood, such as the effects of age or cause of death, there are definite patterns in which genes are transcribed at different times after death. And that, forensic scientists note, could be valuable in estimating the time of death in criminal investigations. So dead men tell no tales, but their transcripts sure might. When RNA's role in the DNA to RNA to protein process was first recognized, it seemed to many like little more than a carrier of information. Today we recognize many roles for RNA, and the list is growing. In fact, RNA transcripts are central players in the rapidly developing field of transcriptomics. With this lecture, we've made our transcripts and scratched the RNA surface. We'll dig deeper in upcoming lectures, starting with the next lecture where we discuss the translation of RNA transcripts into proteins that makes us possible.